Welcome everyone, and thank you for attending Terra Universal's webinar on USP and CGMP cleanroom compliance. My name is Evan Messenger and I work in business development and marketing at Terra Universal, and today we're going to get into the nitty-gritty of the USP 797 and 800 regulations and the CGMP standards, and talk a little bit about how the shifting nature of these regulations impacts both your pharmacy operations and specifically cleanroom design. Before we get into the presentations, let me first introduce you to our panelists. We have Dr. Chris Munoz joining us today. Dr. Munoz is a professor of pharmacy compounding at the University of Southern California. He is active on the policy committee and board of directors for the California Pharmacists Association, and he is the principal of ITL Consulting. Thank you for being with us today, Chris. Chris is joined by Dr. Jesse Martinez. Dr. Martinez is the Vice Dean of Western University's College of Pharmacy and brings with himself 37 years of experience in sterile and non-sterile compounding, as well as pharmacy administration and research. Final member of our panel today is Mike Buckwalter, who is involved with both engineering and marketing at Terra Universal. So, little schedule of events today. The first 15 minutes, Dr. Munoz will be talking about compounding pharmacy cleanroom design as it regards USP and FDA compliance. And Chris will get into a little bit of the regulations and the USP and CGMP seeming conflicts. And we'll talk a little bit about how we see these regulations enforced over the coming years. He'll then be succeeded by Mike Buckwalter, who will be talking for about 15 minutes specifically on clean room design. And then finally, we'll have 25 to 30 minutes to take all of your questions. So before Chris starts, if any questions come up over the next 25 to 30 minutes, please submit them to us through the Q&A chat box, which is in the lower right-hand side of your interface. We will try to answer all of your questions, but if we don't get to yours, we will be in contact with you by email over the next 3 to 4 days to personally answer that question. So without further ado, let me hand the mic over to Dr. Munoz, who will be talking about compounding pharmacy cleanroom design. Take it away, Chris.
So we're here today because the business of pharmaceutical compounding is being held to much higher standards than in years past in regards to both facilities and the processes of production. These standards come from multiple sources, and it's not always simple to understand what a compliant practice looks like. After today's presentation, we can hopefully have a stronger understanding of the regulatory framework and understand the pharmacy's responsibilities. The push for higher standards came about due to the unfortunate events in 2012. Most of us are already familiar with the details surrounding the event of the fungal meningitis outbreak that was a result of the contaminated injectables from NECC. We won't go into too much detail in consideration of our time, but in short, over 60 people were killed and hundreds of people were hospitalized. Needless to say, the public was outraged, and it was the political backlash that resulted in a complete overhaul of the regulatory landscape, both federally and locally, state level, that is. So today our focus is going to be on the regulations that are now in play as a result. You are all probably familiar, at least in a general sense, with the DQSA, Drug Quality and Security Act, the CGMPs, or Current Good Manufacturing Practices, and of course USPs 797 and Chapter 800. But you may not be as familiar with how these affect your pharmacy and its operations. So we're going to examine how these new and revised regulations affect specifically your facility's requirements, whether you're compounding sterile medications, hazardous drugs, or both. We expect this presentation to hopefully save you some time and frustration in the comprehension and implementation of these regulations, because we're going to walk you through the key highlights of each regulatory document step by step so that you can hopefully put the different pieces together in order to see the bigger picture. By the end of this presentation, questions included, hopefully you should be able to make better, more informed decisions when setting up or making changes to your compounding facility and its operations. So rather than providing you with bullet points of information, what we're going to do is take you through chronologically the events and the highlights of the key documents where these regulations are found. So you're going to see the exact language used by the FDA and the USP in black and white. Each of these documents are publicly available online at each organization's website, and we can also provide attendees with the links to all this information as well. But for the purpose of this presentation, you don't need immediate access to them. You'll be seeing some highlights of these documents. So let's begin with the DQSA. So our discussion is going to start at the federal level, where, as you know, in 2013, Congress worked together to pass regulations that define the new category of compounding pharmacies we know as outsourcing facilities. It also grants the FDA oversight of these facilities under Section 503B of the Food, Drug, and Cosmetic Act, or simply the FDCA. Let's take a quick look at the meeting where the implementation of the DQSA was discussed among the FDA representatives as well as representatives from the State Board of Pharmacy. So, quick history lesson. You're looking at the minutes from the 2014 Intergovernmental Working Meeting on Pharmacy Compounding as hosted by the FDA and as attended by many, including representatives from the State Boards of Pharmacy. At the bottom of this page, they acknowledge the passage of the DQSA, which you'll notice mentions both state and federal oversight for compounded human drugs. In regard to oversight, they refer to Section 503A, which has historically exempted pharmaceutical compounders from the regulations intended for the manufacturers, which are GMPs, as you know. And that's nothing new, but rather it's an inclusion and pretty much for the sake of completion, given the history of pharmacy compounding in the bigger picture. This will, however, be important as we move forward in our discussion. The new section in the DQSA, which we've already talked about, is Section 503B, which gives the FDA the authority to regulate outsourcing facilities, as we've just mentioned. In this definition, an outsourcing facility is basically a compounding pharmacy who either actively elects to register as such or who does not fit the definition of a traditional compounder as defined in Section 503A of the same law. Outsourcing facilities will be expected to follow CGMPs for sterile drug products while under federal oversight. That means guaranteed FDA inspections according to those said standards. So what are the CGMPs that outsourcing facilities are going to be expected to follow or are currently being expected to follow? So a few months after that meeting we just discussed, in July of 2014, the FDA published a draft guidance document that would generally explain their expectations for the 503B outsourcing facilities. In this guidance document, the FDA announced their expectations for outsourcing facilities. The key point to understand about this draft guidance is that these documents are only the expectations of the FDA during the interim period until they write a more definitive guidance document. This is critical, as you may know. The FDA has already started inspecting registered outsourcing facilities according to GMP standards and have cited the relevant sections in 21 CFR, which is the Code of Federal Regulations, in addition to violations pertaining to sterile and aseptic manipulations. More details on those can be found on the FDA's website in the section Form 483s for outsourcing facilities. You can take a look there after the fact and kind of see what the FDA is looking at in terms of both federal regulations as well as more detailed sterile and aseptic manipulation standards. But given that we're here to talk about specific requirements for facilities, let's take a look quickly at what this guidance document specifies. So here, what you can see is the FDA's goal overall is to prevent contamination of compounded sterile medications. And throughout the document, you'll see much emphasis on the cleanliness and ISO classifications, which refer to the air particle counts within a clean room. You will notice that the ISO 5 rating is prominently featured in most of their recommendations. This represents the minimum acceptable level of air purity where manipulations of sterile drug products must take place. This is usually achieved with laminar airflow hoods that can be certified to meet these specifications, In other cases, companies may opt to have an entire room or sections of the room certified to be ISO 5 level purity. This can be done by increasing the number of air changes per unit time with HEPA fan filter units, but we'll get more into the considerations for design coming up a little bit further on. If you are an outsourcing facility, you can undoubtedly expect the FDA to be observing your facility and watching for any exposures of the sterile materials to anything less than ISO 5 conditions. That said, the facility will need to be carefully designed to include the ISO 5 critical environment with respect to your specific operational size and scope. And again, we'll get more into depth on this point a little bit further on for the presentation. Now, there are more detailed operational considerations for CGMP that can be found in older FDA documents which pertain to pharmaceutical manufacturers, but this is where the emphasis has been on both paper as well as during inspections with regard to the facility. So this draft guidance we just talked about was released in 2014, and as we go through chronologically, let's take a look at a more recent development in the implementation of the DQSA. So in March of this year, FDA held a third intergovernmental working meeting with the main purpose of harmonizing their enforcement efforts with the state boards of pharmacy. The main disconnect between the FDA and the state boards of pharmacy is that FDA has moved forward very aggressively to enforce the standards in CGMP, whereas the states have passed new laws for compounding, but generally lack the resources and the knowledge to inspect outsourcing facilities within their own borders. However, the states still say that they cannot rely exclusively on the FDA inspections for outsourcing facilities because those standards lack the enforcement of state-specific laws, for example, tech ratios. Because each state's laws and resources significantly vary across the U.S., increased importance is placed on communication between state and federal authorities. Both the FDA and the state boards recognize that there is a lack of harmony in their enforcement practices, and so essentially they resolve to work together on smoothing out this regulatory overlap. As you can see at the bottom of this page, one method of accomplishing this is through cooperation on joint inspections. And who wouldn't want to have a team of joint inspectors coming through? It's so much more convenient and efficient to have a whole team of inspectors show up on your doorstep one day. But not surprisingly, unfortunately, this is something that's already been seen in California. So far, we've primarily framed this discussion around outsourcing facilities, that is the 503B, and you might be thinking at this point that 503B outsourcing facilities have it rough. The truth is, there's more to the story. So let's talk about the other half of the federal law, the 503A, traditional compounding pharmacy, also seen in the eyes of the FDA as everyone who is not an outsourcing facility. So in June of this year, the ASHP released a set of guidelines on pharmacy management that actually included a section on outsourcing facilities specifically. Along with their recommendations for choosing appropriate outsourcing facilities, the ASHP gave an especially timely warning regarding the 503A compounding pharmacies. They took note of the fact that if the FDA has the authority to regulate pharmacies under Section 503B, then why wouldn't they also have the power to inspect and regulate Section 503A traditional compounding pharmacies? Well, that's exactly what happened. Along came the FDA's final guidance document for Section 503A in this past month in October, and that's what they said. So if we get into this document, right away on page 3, the FDA makes it very clear that although they expect the state boards of pharmacy to take the lead on regulating any non-outsourcing facilities, they still retain the authority to enforce the provisions of the FDCNA for Section 503A traditional compounding pharmacies. So on a side note, you might be a little confused by the phrase at the top of the page, which stipulates that these are non-binding recommendations. However, closer look will reveal that these non-binding recommendations are actually references to 21 CFR and, of course, all the various sections that are definitively written into law. Furthermore, these recommendations specifically outline each of the possible enforcement actions that the FDA can take against violations of the FDCNA. So you should also note the language used towards the bottom where the FDA defines the possible violations using very broad and subjective terms such as insanitary conditions and may have been rendered injurious to health. What this does is it grants the FDA a large amount of discretion in its inspections, and unfortunately it leaves very little wiggle room for compounding pharmacies found to have any violations. So to quickly recap about federal law, to frame everything properly in your mind, we now know that the FDA has the power to inspect both 503A and 503B compounding pharmacies. The question, unfortunately, is not if, but when the FDA will show up. And although the stricter standards of CGMP will only be enforced with outsourcing facilities in most cases, we also know that the FDA has very flexible powers and a lot of discretion in how they also define violations against 503A traditional compounding pharmacies. And although the draft guidance documents indicate that they operate on a risk-based schedule, in many cases we're not seeing a risk-based schedule followed, and we're seeing surprise visits to 503As in general. So we can safely say that the pharmacy is responsible for standards for 503A, and if you're a 503B, of course, that's very clear-cut. So now let's compare the FDA's standards with those at the state level. And for the purpose of this presentation, we will refer to the USP standards as the state standards. Since the publishing of 797 in 2004, the state boards of pharmacy many state boards of pharmacy have adopted USP to some extent. But as we've seen from the intergovernmental working meetings, the FDA and state inspectors have not yet reached a common ground in terms of their enforcement practices. So in an effort to catch up with the stricter federal regulations and create a definitive standard, USP is close to completing an overhaul of 797, which includes breaking out the section on hazardous drugs into its separate set of guidelines, which is, as USP 800. And for our purposes, we're going to focus in on the changes that affect the facilities and the equipment. So what you're looking at right now is the proposed draft of the newly updated 797 as posted on the USP website for public comment. USP has completely reorganized the chapter and has also changed how they classify the risk level for CSPs, or compounded sterile preparations as they define it. The newly classified categories are Category 1 and Category 2, which as you can see in front of you. The main difference is that these two categories, for Category 2, must be prepared in a cleanroom environment. Without the ISO classified cleanroom, the beyond use state of all CSPs is going to be restricted to a maximum of 12 hours at room temperature, or 24 hours under refrigeration, and that would be classified as Category 1. You'll also notice down there in point number 3 that they have removed all references in USP 797 to hazardous drugs, and instead now refer exclusively to USP 800. So the draft version of 800 has been posted on USP in its current form since about October of last year. So we're going to assume, for the purpose of our presentation, that most of us listening in today are somewhat familiar with its provisions, and we will address the major areas of concerns and just the major areas. So from the get-go, we can already see that this draft version of 800 will need to undergo another revision because they're still using the low, medium, and high risk categories as opposed to the new Category 1 and Category 2 for CSPs, which we just looked at. A timeline for finalizing USP 797 and 800 was already hazy, but with both of these documents still undergoing public comment, it's difficult to predict when these documents will become enforceable. The best approach we can take in the meantime is to focus on the main concepts and clarifications that will be unlikely to change given a revision of the language. In this case, we should note the elimination of a major exemption for low-volume hazardous drug compounding. USP is making it clear that all hazardous drug compounding must take place in a negative pressure room, whether it's a clean room or a segregated compounding area. To further complicate matters, hazardous drugs will also need a dedicated negative pressure storage area, with a few exceptions noted. You can see here the specific storage exemptions in the first highlighted sections on lines 124 and 125. Because the language here is very specific, we think it's unlikely to change significantly in any final revisions. At this point, though, we are still waiting for their response to public comment period, which ended back at the end of May of this year. But among the experts who have participated in USP's revision process, the consensus generally is that the final version of 800 will not be much different from this draft, except for the change in the classifications that we have already noted. So to be safe, if you're planning on compounding hazardous drugs, you should definitely make sure that you can install the ventilation system that will be able to achieve negative pressures in both the compounding room and in your biological safety cabinet, as well as any negative pressure storage rooms that you may need. All of the negative pressure ventilation also has to be drawn through a HEPA filter before it is vented to the outside air, which can certainly pose an engineering challenge. So we've looked at the federal regulatory responsibilities for the pharmacy. We now know who you will be required to respond to, and we've looked at the emerging state level standards as well. So it's safe to say that there's multiple standards at play. There's multiple levels of considerations here. And with that said, we're going to continue the discussion with regard to the specifics on the facility standards. And I'm now going to turn the mic back over to Evan.
Thank you so much, Chris. Very informative. Before I give the floor to Mike Buckwalter, let me first reiterate, if you do have any questions for us, please submit them through the Q&A chat box located in the lower right-hand corner of your interface. So I'll give the floor over to Mike. Mike is going to take a very engineer-centric approach, and he's going to talk about specifically clean room design and the impact of the USP 797, 800, and the FDA CGMP regulations on how your clean rooms now need to be designed to ensure that your facility stays within compliance. Take it away, Mike.
Okay, well, thank you very much. We will be, as Evan mentioned, we'll be focusing on how some of the issues that Dr. Munoz just reviewed for us affect the specific environmental design and control considerations involved in clean room design, and specifically the recommendations that Tara offers for the designs that we promote. And let's start by looking at the question of who controls, what these controls are and oversees them. And we'll run pretty quickly through some history on the development of clean rooms. Many of us first learned to classify clean rooms or discuss clean rooms in terms of the old specifications developed by Fed Standard 209 50 or so years ago. Those were replaced about 2001 when the International Standards Organization, their recommendations came into force and Fed Standard 209 was suspended. So the ISO 34567 replaced the old Class 1, 10, 20, so forth. We also have European GMP guidelines in effect, and these have been influential in the United States as well as Europe, and a different classificatory system in Britain. So there are a number of bodies out there, but the ISO standards are increasingly the predominant recommendations now. And interestingly, ISO doesn't have much to say about specific design considerations. They're concerned about the final result, which is particle levels. So they say a great deal about how to define ISO levels in terms of particle counts and how to measure those particles. And of course, they came up with the chart that's represented on the slide you see right now. But they don't tell us much about how to achieve those counts. And for that information, people usually have to refer to other information sources. Now, one of the environmental control considerations in a clean room design, of course, is laminarity. And laminarity is very difficult to demonstrate in a large clean room. Typically, smaller enclosures, the primary engineered controls that are referred to in the USP specs, are carefully monitored to make sure that they maintain unidirectional airflow, all air moving in the same direction at the same speed. But it's much more difficult to do that on a large room basis, and particularly in a clean room classification that's not as strict as an ISO 4 or 5. So laminarity is more of a concern on with smaller enclosures. Another key consideration in clean room design, of course, is the air change per hour rate. And for these numbers, or for a reference on ACPH numbers, people often look to IEST, which has been the major resource for some of these specific recommendations. And as you'll notice here, IEST offers fairly broad ranges for each of the ISO classifications. Looking at ISO 7, for example, which is a level commonly called out by the USP specs, that range varies from 60 to 90 air changes per hour, quite a broad range. And in the USP spec itself, the number that's called out is 30 air changes, a much lower number. So it's striking how much variation there is in the area of air change rates. Balancers often insist on much lower numbers, and when you move into the CGMP world, that's where most often an inspector is going to look at the IEST numbers, which are much higher. So that's the first point to make here. Generally, in our recommendations, we're looking at IEST. So you may see a room design that goes far above the 30 air changes recommended for ISO 7. We're trying to put you somewhere in that 60 through 90 range. Another issue involved related to air changes, of course, is the ceiling HEPA filter coverage. And there are simple calculations you can perform to see what portion of the ceiling should be occupied by HEPA filters in order to achieve the recommended particle counts and air change rates. I won't go into the numbers here, but they're there for your reference. One point I wanted to make here is that the ceiling coverage is, of course, since it's a volumetric issue, is related to ceiling height. So in a conventional brick and mortar clean room with a higher ceiling, you'll need a higher air change rates than you would in a typical modular room that might have a lower ceiling. And then the next consideration involved here is air speed. Typically, certifiers for a clean room in the ISO 5 range tend to use 90 feet per minute as a reference point. That's become kind of the magic number for air velocity. Technically, in an ISO 7 or an ISO 8 room, we do not need that quite that airspeed, but in the cleaner rooms and the environments that might come under CGMP, we're looking to maintain somewhere around 90 feet per minute. Other considerations involved in clean room design include in-use considerations, the number of people that might be involved in a room. The personnel count also affects, if it's a room that involves recirculation, it affects the amount of makeup air that has to be introduced into the system. Of course, if we have heat generating or particle generating equipment, then we may also need to move to the higher end of an air change range than if we didn't. And then other considerations involve placement of exhaust vents. Ideally, we want them located throughout the perimeter of the room, if possible, to try to maintain and optimize that unidirectional airflow. Ceiling height, we already mentioned, and then considerations like entryways, ducting, and other issues like that. And by the way, entryways typically leak air. Even a fairly well-sealed door will leak 75 to 100 CFM or more, depending on how well sealed it is. So those are all things we have to think about when we're designing a room and figuring out how many ceiling fans we need to install. So how does this shake out in terms of specific USP room considerations? Well, in the typical USP 797 sterile compounding room, we are looking at a positive pressure system. We are trying to maintain typically ISO 7 or a little bit better. So we'll have our ceiling filter fans that are directing the airflow downward. Air is exiting the room along the near the floor, along the perimeter of the room, as you see in the illustration. And in a regulated industry, we're generally going to want to provide some sort of automated control system so that we can dynamically manage airflow speed, both to ensure uniform air speed and also to maintain proper pressure levels. And you'll notice the bold-faced bullet. We typically are out to maintain a pressure differential between 0.025 and 0.05 inches of water column between rooms of differing ISO levels. So in a typical USP 797 room, we need to maintain that differential between the buffer room and the anteroom. The next consideration in a negative pressure room, a USP 800 containment room, the cleanliness requirements hold that we've just reviewed. So we have our ceiling fan filters. We're maintaining the ISO cleanliness level that's prescribed, but now we need to pull a certain amount of air to maintain that overall negative pressure with respect to the outside environment. And typically that needs to be a minimum of 0.01 inches of water column negative pressure. We're generally going to try to recommend a little bit more than that so that we have a margin of safety. But this does add to the expense of a room like this, because now we need to provide some provision for removing that air. Typically that will be through exhaust ducting installed near the base of the room right outside the buffer area, as you see in the illustration. And then that air will be directed typically upwards and tied to an exhaust system, which is typically roof mounted. And in the rooms that we're recommending and that we provide to our customers, we'll provide a means of connecting that ducting system to a roof ventilation system. And we would work with a local contractor or whoever might be installing that roof fan. But and as well to help specify the size of the necessary fan. Typically, this would be a centrifugal fan with the capability to pull all of the air we're putting in the room plus 30 or 40% additional air, just to compensate. Well, first to provide the negative pressure and to compensate for any static pressure that may develop in the exhaust line. Now, in a sterile-to-sterile hazardous drug compounding situation, we would be recommending a number of room designs. The one that you see here is a very versatile layout, space-efficient, that also provides capability for both hazardous drug and non-hazardous drug compounding. So as the USP spec prescribes, you would enter into an ISO 7 anteroom, and you would move either into the negative pressure hazardous drug room, which is represented here on the left side, or into the positive pressure USP 797 room for non-hazardous drug compounding. And the specifications only require ISO 7 for that positive pressure room. We go a little bit cleaner in order to maintain the air pressure gradient that ISO as well as USP would require. We need to have positive pressure with respect to the anteroom. So we're going to install an extra fan filter unit or two to provide that. Fortunately, our designs are very, very modular, very flexible, and these room classifications can be modified at fairly low cost simply by adding another fan filter. On the left side in the hazardous drug containment room, as you can see, we're pulling air out of the room into those orange exhaust plenums, which are located near the floor of the room, and then exhausting that room to the outside world. On another variation of this would be a room that just includes the hazardous drug compounding and the anteroom. So in this case, this would be a facility that would not be doing non-hazardous drug compounding, but the basic design is essentially the same. In a non-sterile to sterile compounding scenario, hazardous drug compounding, we are now going to increase the cleanliness levels throughout the facility, well, particularly in the hazardous drug area. And we're doing this because we're sensitive to some of the FDA enforcement practices that Dr. Munoz just outlined for us. We're much more likely here to be looking at a 503B scenario or a non-sterile to sterile hazardous drug scenario where FDA is going to increase its requirements and really put us under CGMP design considerations more than the USP considerations that many people have followed up till now. Fortunately, the expense in doing so is not extreme. We'll add an extra fan filter or two, but that's not necessarily an expensive proposition. One of the main cost considerations here is the more air you put in, the more air you have to pull out. So often we'll have to size those exhaust ducts a little bit larger and then, of course, provide or recommend a much larger exhaust fan to ensure the negative pressure that we need to remain within spec. So these rooms are typically 2 to 3 times more expensive than the smaller rooms. On the USP 797 side, you can have a room like this designed and built for under $20,000. Some of our rooms start as low as $15,000. You need to triple that cost at least when you're looking at a hazardous drug facility because of the containment provisions, the need to provide a ducting system, the fan, and so forth. This slide represents the same type of a room, but here without the positive pressure compounding room. So we have our negative pressure, again, ISO 5 for a 503B facility, our negative pressure room, and then the anti-room leading into it. Temperature and humidity are other considerations in cleanroom design, and very briefly, we typically want to go 5 to 10 degrees Fahrenheit below ambient just for the comfort of the operators who will probably be working in cleanroom coveralls, which are quite warm. If you've ever been in that situation, you know. Relative humidity typically needs to be maintained in the 40 to 60% RH range. So typically, if we're installing a clean room in an existing facility, we're already within that range, but sometimes we may need to add or pull humidity to remain within spec. And another consideration in especially a regulated industry like this is that we want to provide some automation in the control systems. And for a USP 797 room, we have rooms that operate simply with analog magnehelic gauges to monitor positive pressure, but most often we are looking to provide an automated system that monitors and gives you fan control from a central console. In our 800 rooms, the negative pressure rooms, these are built into the unit. This is much more critical when you're managing negative pressure, to be able to dynamically adjust your fan speeds to maintain those pressure gradients that ISO and USP call out. So in a situation like that, we would be installing sensors, typically room pressure sensors, possibly also RH, temperature, even particle counters in each room, and then a control system that lets us ramp up or down our fan speeds in order to maintain the prescribed pressure levels and air change rates. Summary of cost considerations then. Kind of summing up everything we've said. On the modular cleanroom side, we reduce both cost and installation speed, and these are the rooms that Tara is involved with. We do some room conversions, and we can take a standard existing facility and do a conversion to meet cleanroom requirements, but most of our installations, especially for compounders, would be modular rooms that we can install within an existing facility. And as I mentioned, these rooms would start around $15,000, not including installation, but they're typically much less expensive than a comparable brick and mortar conventional room would be. And because they can be installed into an existing facility, typically we can leverage existing HVAC systems so we don't have to get into the cost involved there. As I mentioned, when we move into a containment room, our second point on this slide, we're looking at the additional expense of the exhaust fan, the exhaust ducting, and a more robust automated control system that is going to issue alerts and certainly provide the monitoring necessary to maintain the conditions. An advantage of those systems also is that we can log those data so that you have a traceable storage history of all the, over the course of your certification period of room performance 24/7. In a case like this, with these automated systems, there's no need for personnel to manually log those pressure levels, which USP 797 requires. So this makes that the burden of proof and documentation much easier to meet. Air conditioning, as I mentioned, adds a cost. As much as possible, we're trying to leverage existing systems to keep room costs low. And these automated systems also save when it comes time to certification, to perform certification. A room with automated controls obviously is much quicker and easier for a certifier to audit. These controls also provide energy savings. So these are all things that, ways that we can help contain costs with a modular room that employs these automated systems. And at this point, I'll turn things back to our moderator, and we'll address your questions.
Thanks so much, Mike. And thank you to everyone who pre-submitted questions during the registration process. We'll answer your questions first. In fact, we have prepared a couple of slides in support of our answers to those questions. But if any questions do arise during this time, do submit your questions to us through the Q&A chat box. So before we answer the questions, let me again introduce our panelists who will be answering your questions, the Drs. Munoz and Martinez and Mike Buckwalter. And I may jump in and answer a question or two myself. So let's get to our first question. The September 2015 USP 797 proposed revision states that all compounding of Category 2 CSPs must be performed in an ISO 8 cleanroom if using an ISO 5 isolator. We're currently compounding in an ISO 5 isolator located in an unrated space. Do I need a cleanroom? And again, a quick aside, the new USP 797 revision has recategorized CSPs from low, medium, and high risk to Category 1 and Category 2. The answer to your question is not yet, so long as your isolator can be certified to ISO 5 under dynamic conditions. For now, you're okay. But once the revised USP 797 becomes official, you will need an ISO 8 room with your ISO 5 isolator installed to do all compounding of Category 2 CSPs, so long as you're not compounding under the 12-hour or less beyond use date exemption. However, if you're a 503B outsourcing facility, this requirement may be enforced sooner as the FDA finalizes its CGMPs. Now, you may be thinking, an ISO 8 cleanroom, that doesn't really jibe with the spirit of the regulations, and that's true. We recommend for you in this instance, install your ISO 5 isolator within an ISO 7 buffer room with an adjacent ISO 8 gowning and staging room. And this gives you a little bit more facility and operational flexibility should you decide in the future to proceed with compounding, say, hazardous CSPs, or if you are working within the beyond use date exemption and you start compounding CSPs that are no longer under that exemption. This gives you some facility flexibility. So that's our recommendation in those instances. This allows us to segue very quickly into the different types of primary engineering controls. Now, I think you're all probably quite familiar with glove box isolators, and these isolators will maintain ISO 5 conditions within the work area and also provide a secondary level of containment should a fan become disabled. And because of that, FDA inspectors would rather see an ISO 5 glove box than an ISO 5 laminar flow hood. But you may use an ISO 5 laminar flow hood for compounding of non-hazardous CSPs. Now, a laminar flow cabinet maintains the same ISO 5 conditions within the work area, but the air is passing the operator as it leaves the hood. So it's not appropriate for compounding of hazardous CSPs. Now, if you are compounding hazardous CSPs, the design that we would recommend for you would be a biosafety cabinet, and specifically a Class 2 B2 biosafety cabinet. Now, this may, on the outside, look like a laminar flow hood. It does maintain ISO 5 conditions within the work area, but rather than the air passing the operator as it leaves the hood, it is diverted, filtered, and ducted through the back of the unit and can be brought into an air scrubber or brought directly out of the facility. This keeps your operator safe if you're compounding any CSPs that are listed on the NIOSH list. The fourth category is a fume hood. You may have run across fume hoods from time to time. Fume hoods are not recommended for compounding of sterile to sterile CSPs. Yes, the operator is protected from exposure to CSPs, but the intake air is not filtered, so your CSP is prone to contamination. Great question. Our second question, can we transport our mini bags into and out of our clean room using a cart? If not, what sample transfer options do we have? And I think I'll pass the mic over to Mike Buckwalter, who can answer that question.
Yes, the carts, my understanding is that carts should not be moved back and forth between the ante and buffer area. And I think this is a rule of thumb that, that dates way back and has been addressed in USP 797. And the idea is we don't want to violate that, that sort of boundary cleanliness layer that we established between the ante and buffer area. So typically, when you're moving a cart into a buffer area, it should stay there for the duration of a compounding shift. So it's not a good way to move product in and out of the buffer area in a high workflow environment. And that's partly because the cart would need to be decontaminated every time it moves in and out, and that's an involved process that involves many steps, and generally that would become cumbersome. So this question was sent to us in advance, so we put a slide up here to cover one option that we provide in a high workflow situation like that, to install a pass-through chamber. And as long as that pass-through chamber is interlocked, it provides an easy way to move compounded material from the buffer area to the outside world. And the interlock system, ours are typically use an electronic system or a mechanical system that would ensure that you can't have cross-contamination. Only one door at a time can be opened. And that could be installed between the buffer and the outside world, or between the buffer and the ante chamber or the ante room, depending on your workflow. Typically, though, it's going to be between the buffer and the outside world, so that you can pass compounded materials out of the clean room. Oh.
Thank you, Mike. Our third question comes from Gerald. Gerald asks, what is the smallest by square footage feasible USP 797 compliant clean room? And we get this question often in pharmacies that are strapped for space. Mike, can you take this one?
Sure. The practical limitation on this is the requirement to have the ante room and the buffer room. And the buffer room itself needs to be large enough to accommodate a reasonably sized compounding area. Traditionally, most people regard that as, at minimum, a 48-inch linear inch workstation, whether that's a laminar flow, primary engineering control, or a biosafety cabinet. To be able to position that and have those 48 inches as a space to lay out the preps, and then have some working room, that you're probably going to have difficulty sizing a room much smaller than about 6 feet by 8 feet, which is our smallest facility. That would be roughly split between about a 40 to 60% ratio between the ante room and the buffer room where you have the engineering control. So 6 by 8 roughly, 48 square feet would probably be the recommended minimum.
And thank you, Mike. Our third question, what is the necessary minimum air change rate for a USP 797 and a USP 800 clean room? Another engineering-centric question. Can you answer that one, Mike?
Sure. I think we touched on that already, and depending on whether you fall under a FDA scrutiny as a 503B facility, in which case you would probably want to maintain IEST recommended air change rates, or you're a conventional non-hazardous drug compounder and you can get by with the 30 air change per hour rate that we saw on the slide a little bit earlier. And one note I'd like to mention here is generally you're going to be visited by a certification company which will document all of your environmental conditions, and it's important to work closely with them. So when someone is visiting your facility and determining the air change rates, if you think you need 30 as a conventional, say, USP 797 compounder, you want to make sure that they're documenting that room in use. So you have dynamic situations, people moving around, doors opening and closing, or if it's a tighter scrutiny under possibly FDA regulations, that you have them do likewise and target the high end of that IEST range, so that there's no question when whether it's an FDA or a state board auditor comes to inspect those results, that they see that this was in use and that you comfortably meet whatever requirement applies.
Thank you, Mike. And before we let you rest your voice, how about one more engineering question? What design features besides ISO cleanliness distinguish a USP 797 from a USP 800 clean room and from other clean rooms?
Okay, well, very quickly, we've touched on some of the design considerations, airflows, air change rates, and those sorts of things. Beyond that, I would highlight things like things dictated by protocol. So in most cases, you want hands-free operation. Doors should be, if they're swing doors, they should be able to be manipulated without using hands. If cost allows, you could use an automatic door with a no-touch panel, so that you're not contaminating gloves or hands as you're entering the ante room or the buffer room. Some other issues, we're always going to want to cove our floors to the walls so that the floors can be easily cleaned. Terra's modular clean rooms for this industry all feature very smooth interior panels. You're going to have to demonstrate that you're meeting the sterilization protocol dictated by USP, so you don't want internal irregularities in surface. You want everything very smooth. It needs to be compatible with alcohol or whatever biocide chemical that you're using, so a chemically compatible smooth interior surface. And then, as I mentioned earlier, ideally you're going to have an automated control system that documents your internal conditions. So that's something that I think is particular to FDA and especially to these compounding rooms. And I'll mention also there's a little bit of controversy out there about pressure analog versus digital pressure gauges. We actually provide both, and a lot of people like the analog pressure monitors because you see that the room is live. You see a needle quivering dynamically in response to what's going on. The digital systems provide much more versatile and traceable conditions, but you can walk up, take a look, and see right away that you're maintaining a pressure inside the room. So that's another feature that we'd want.
Thank you, Mike. Well, I think the audience is pining for an answer from Dr. Munoz. Question number 6, can we prepare low and medium risk CSPs within the same ISO 5 laminar flow hood?
So, in short, yes. As long as all of the ingredients are sterile, there's typically not a problem with low and medium risk being done in the same hood. What you probably want to avoid is doing them simultaneously, providing that you have a multi-operator hood. That would be a little bit risky. And of course, the exception would be with high-risk compounded sterile preparations, where of course you're going to clean in between each batch, and you would sterilize essentially all of your materials, both before, in between, and then after.
Thank you, Chris. Now here's a question that I see often. We have a refrigerator filled with reagent kits located in our positive pressure ISO 8 anteroom. Are we within compliance? Should our storage area be located within a neutral pressure space? What's the answer to that, Chris?
Well, typically because refrigerators are going to be particle generators, they're typically placed outside of the ISO classified areas. It's not to say that it's impossible to have a refrigerator in an ISO 8 anteroom, but it does make it much more difficult to achieve your ISO 8 certification, especially during operation on a daily basis. To be clear, a refrigerator should never be placed in the ISO 7 buffer room, and if we're talking about hazardous drugs, you're going to need stored away from other inventory. But they only recommend a negative pressure environment. There's no ISO classification specifically. It's just a negative pressure room with at least 12 air changes per hour.
Thank you, Chris. This question is from Matthew. Will the 12-hour beyond use date exemption be affected by the USP 800 FDA regulations? Chris, do you know the answer to that?
Yeah, no. USP 800 and CGMPs do not have any exemption for the 12-hour beyond use date. It's USP 797 that's going to allow for the ISO 5 primary engineering control, as they refer to it, to be located in a segregated compounding area, provided that you do assign it a beyond use date of 12 hours or less.
Here's an HRT question from Brett. He's doing USP 800 non-sterile compounding of biohormone replacement therapies. What type of clean room does he need? Chris, could you answer that?
So USP 800 will require the negative pressure room and negative pressure biological safety cabinet that we've mentioned. However, the exemption is if the hazardous drug is classified as reproductive risk only, then the cabinet air can simply be recirculated into the room, provided it's double HEPA filtered like we mentioned at the end of the presentation. The room air will definitely need to be externally vented. However, neither of these need to achieve any sort of ISO level classification. Again, it's simply the negative pressure and the air changes that are required by USP 800.
Thank you, Dr. Munoz. And another question that is right up your alley. David asks, do you recommend switching the most common disinfectants in my lab from IPA or bleach or hydrogen peroxide? Does the USP or DQSA recommend other disinfectants? I mean, we always see 70% IPA.
In terms of disinfecting, there's not really any specific requirements with respect to 797. The main idea is that you want to rotate them. 797 does include a list of alternative disinfectants along with their cleaning properties, but either of those should be fine, providing you're following the general kind of good practices of rotating your disinfectants and documenting your cleaning practices. The FDA's GMP guidance requires similar procedures as 797.
Thank you, Dr. Munoz. Jeffrey asks, when is room laminar airflow a requirement for a GMP pharmacy? Is it only for compounding operations? Are there other operations that may require laminar flow air? Chris, could you take that question?
So clean rooms are required by USP and CGMPs to have unidirectional airflow, and any turbulent air should be avoided. Actual laminar airflow should be more strictly observed in the ISO 5 compounding area, which you can get certified by any sort of certification company or qualified technician using various tests. The smoke test is one of a few tests that can be used to verify laminarity and unidirectional airflow.
And our final pre-submitted question, Marcy asks, is a sporicide required for terminal cleaning? What is the recommended procedure for wiping down material prior to entering the anteroom? Chris, do you have an answer for that?
So the revision of 797 provides probably the most clarity on this topic, and they recommend using a sporicidal agent weekly to clean the ISO classified surfaces. Shipping containers and cartons aren't allowed in the anteroom, but the compounding supplies only need to be wiped down with isopropyl alcohol, or preferably sterile isopropyl alcohol, provided they're going into the buffer room and into the critical ISO 5 environment.
Thank you very much, Dr. Munoz. Now, we just have a couple of minutes left, and we'd like to answer a few additional questions that have come streaming into us. Again, we'll be in contact with you by email if we haven't answered your question, but let me send a question over to Dr. Martinez. Ricardo asks, what is the panel's opinion on when this draft for 503B will be finalized? Jesse, could you answer that for us?
I'd be glad to. That's a crystal ball question. I think more importantly, we can say that since this draft has been out by the FDA for almost a year now, I would say the very soonest they can get this finalized and enforced would be sometime before mid-year next year. That would be the quickest, I believe, in my opinion. But I can see this this draft being a very large impact to the industry, so it may be even stalled even longer. But I recommend that, Ricardo, you monitor the FDA website to make sure that you understand who is working on the draft and when those meetings are occurring. The website will provide that information for you. In addition, there is an executive director for the International Compounders right in Washington, D.C., who spent a lot of time with this regulation, and his name is David Miller. So if you go on to the International Compounding Society and take a look at their website and see how they're monitoring the day-to-day on these particular drafts. Chris, do you have any additional comments?
Pretty much nailed it. It's anyone's guess right now, and your best bet to stay informed is to take a look at what Dave Miller is doing with his group and to follow along and participate in not only all their monitoring, but all their efforts as well.
Yeah, thanks so much, Ricardo. Great question. Well, I think we'll be signing off and letting you get on with your precious time today as we get into late Friday morning and late Friday afternoon on the East Coast. So we will be in contact with you if we weren't able to answer your question during the webinar. After the webinar's ended, if you do have any questions that come up, please direct them to me. My email address is on your screen. It's evan@terrauniversal.com, and we'll work to have an answer to you within the next 3 to 4 days. And also, during that time period, you'll be receiving an email from us that contains both a link to the video recording of this webinar, and also it'll contain a PDF copy of Terra Universal's white paper entitled Designing Your Compounding Cleanroom to Meet FDA and State Board of Pharmacy Guidelines. If you have any additional questions on these regulations, a couple of good resources for you. The ASHP's website has a number of white papers that would answer a lot of questions, and also Terra Universal's website, terrauniversal.com, has videos, white papers, and cleanroom schematics that may answer some of your questions. If you'd like to speak to a live cleanroom specialist, please give us a call at 714-578-6000. I've been your MC, Evan Messenger. Take care, everyone. Thank you so much for attending, and have a wonderful weekend.